In this section, we're going to talk about enzyme-linked receptors. In this case, instead of there being a G protein, for example, the intracellular domain of whatever the receptor is either linked to an enzyme or is itself an enzyme. The ligand is going to bind to the receptor, and then the enzyme gets activated and can do its enzyme business. So here's an example called tyrosine kinase receptor. A kinase is an enzyme that transfers a phosphate from an ATP to another protein. So we've talked in a lot of cases about removing an ATP. There's actually an enzyme that has to do that reaction every single time. And it's called a kinase, but there's a bunch of different kinases all over the cell. All right, so what we've got is two signaling molecules here in this tyrosine kinase receptor that sit next to each other. And on their extracellular domain, they've got a binding site for a signal molecule. And both of them have to bind to signal molecules for the whole thing to work. When that happens, they actually move toward each other, which they can do because remember the cell membrane is a fluid mosaic. Things can move around within the membrane. And now they have created a dimer. They have dimerized, which means they just created a two-part thing out of two separate things. Okay? A disaccharide is also a dimer. The tyrosine molecules, which are part of the intracellular domain of this receptor, then get phosphorylated. So that requires 6 ATPs, each of which give up one phosphate, produce 6 ADPs, and now the receptor is activated. Now, other proteins called relay proteins can be activated and cause a response. So these inactive relay proteins bind here, probably get phosphorylated, and then do a cellular response. All right. The response can be kind of complicated, and it's often called signal transduction, which is a series of reactions. So this is one from your textbook. Here's one showing a growth factor binding and to a growth factor receptor. And it creates, this like 4 or 3-step thing that phosphorylate or hydrolyzes GTP to GDP. That then starts off another couple of reactions that finally create something that goes into the nucleus to stimulate transcription and then creation of proteins. We also call these things signaling pathways. So signal transduction or signaling pathway is a description of how the signal gets from one point to wherever it's finally going. Here's what I want you to understand. Most pathways don't look like this. They look like this. This is not something you need to learn. This is the actual pathway of an insulin responsive cell, so a muscle cell or a liver cell, to the presence of insulin. So insulin binds to a receptor here, which causes a bunch of things to happen, which eventually causes this to open a transporter that allows glucose to be transported into the cell. Okay, so when we talk about signal cascades, I just need you to know that it's not usually like 1, 2, 3 steps. It's usually something like this. So please respect the microbiologists and the biochemists who figure this stuff out, because as far as in my brain, this is magic. I can't do this. Anyway, that's signal pathways. We are going to talk about a couple simple pathways. All right. The other thing that often happens in signal transduction is amplification, meaning literally that a small signal is amplified into a larger signal. So here is a G protein linked receptor that binds to adrenaline. Adrenaline is actually a couple of different molecules, epinephrine or norepinephrine. It doesn't matter which one this is. here we bind to the G protein. The subunits break off, and then the subunits are going to bind, or the alpha unit in this case is going to bind with another enzyme, which is going to phosphorylate and create another molecule, except it doesn't do it once. This one signal here of the adrenaline molecule can cause this part to be done tens, hundreds, thousands of times within the cell. As long as this receptor stays here, this process will keep happening. So there are two things that can turn this signal off. One is this disconnects, and the other is that these final messengers are broken down. Not final messengers. These are called second messengers. And in this case, the second messenger is cyclic AMP or CAMP or CAMP. Yes, you need to know about cyclic AMP. Okay. So one of the reason you need to know about cyclic AMP is that it is very often involved in pathways that end up with the transcription of specific genes, or basically turning a gene on and creating the proteins encoded by that gene. All right, so here's what cyclic AMP is. You start off with actually an ATP molecule. It binds with adenylyl cyclase, which actually removes two phosphate groups. See, two phosphates. It's pyrophosphate. I'm sorry, I said phosphorylate, and again, I meant dehydrolyze. Sorry. So when you remove two phosphates from ATP, you get AMP. And it's cyclic because it's going to go through this process a million times, and so it's CAMP. The CAMP is then going to activate another protein called protein kinase A, or PKA. So this is PKA here. Here is 4 CAMP molecules. So this is PKA in its inactive form. It has catalytic and regulatory subunits. The CAMP comes by, and when there are lots of CAMP in the cell, the CAMP binds to the regulatory subunits in these 4 binding sites that activates the protein kinase A, which releases the 2 catalytic subunits. They then go on to catalyze, because they're enzymes, other reactions. In this case, the phosphorylation of a specific protein. When AMP or CAMP levels are low, the CAMP is going to let go of the regulatory subunits, and the protein kinase A is going to go back together into one solid thing. Okay, so this is again a cycle that's going to keep happening of this being activated and then deactivated, and activated and deactivated. Okay, that cycle happens this way. So we have the inactive PKA. Okay, CAMP binds to the regulatory units, lets go of the catalytic units, and then there is another enzyme called phosphodiesterase, which breaks down CAMP. The way it's going to do that, it's actually going to bind to PKA, change the shape, cause a change on AMP. You do not need to know the specific. And then it lets go of the regulatory subunit, and then the regulatory subunit binds back on. Okay, so there's an enzyme that creates CAMP, and there's an enzyme that breaks down CAMP. Okay, so every step of the pathway has to have an enzyme to make it happen, because every step is a chemical reaction. And in biological systems, we control whether reactions happen or not by having reactions that have to be catalyzed by an enzyme. It's actually kind of cool. All right, I'm going to stop there. I'm going to do one more section with signaling in single-celled organisms, and I'm going to introduce you to one of the cutest organisms on the planet.